Welcome back, everyone. Today we're going deep into the world of, well, tiny molecules.
It's going to get pretty molecular, yep.
But don't worry, it's fascinating stuff. We're talking about how these tiny molecules can be like specialized keys.
Yeah, keys that can fine-tune how a receptor works.
It's like having a key that, I don't know, not only opens a door, but like adjusts the temperature or dims the lights inside.
Exactly, and that's what we call biased agonism and allosteric modulation.
Okay, those are some pretty big words.
Yeah, they can be a bit intimidating, but the concepts themselves are pretty straightforward once you break them down.
So let's break them down then. First up, receptors. Can you give us a quick reminder of what those are all about?
Sure, think of receptors on cells as like gatekeepers. They receive signals from outside the cell.
Gotcha. And then they trigger a chain reaction inside, right?
Right, and for a long time we thought these receptors were kind of like simple on-off switches.
Signal comes in, flips the switch, boom, cell response.
Exactly. But it turns out it's a lot more nuanced than that.
Okay, so then what is biased agonism?
So with biased agonism, you've got molecules or ligands that can activate the same receptor but trigger different effects downstream.
Different effects. But how? I mean, if they're activating the same receptor.
Well, it's all about the shape of these molecules and how they interact with the receptor. You see, when a ligand binds to a receptor, it kind of induces a change in the receptor's shape.
Like a handshake.
Yeah, kind of. And different ligands, even if they bind to the same receptor, might induce slightly different handshakes, leading to distinct downstream effects.
So those tiny little molecular details that matter.
Absolutely. And allosteric modulation takes this concept a step further.
Okay, how so?
Well, allosteric modulators, they bind to a different site on the receptor, not the main binding site where the natural ligand usually goes.
So they're not competing for the main spot. They're finding their own little niche.
Exactly. And from there, they can either boost or dampen the receptor's response to its natural ligand. It's like having a volume knob. You can fine-tune the intensity of the signal even if the main switch is already on.
Okay, I think I'm starting to see the difference here.
So biased agonists are like specialized keys. They unlock different functions within the same room.
And allosteric modulators are like dimmer switches.
Right.
Perfect analogy. But to really get why this is important, let's look at a little puzzle.
All right, I love a good puzzle. Hit me.
Let's talk about heart muscle cells.
Mm-hmm.
Cardiomyocytes, they've got these receptors on their surface, beta-1 adrenergic receptors and EP4 receptors.
Okay. And what do these receptors do?
Well, when they're activated, they both lead to an increase in this messenger molecule called CAM-MP.
CAM-MP, yeah, that's involved in tons of cellular processes, right?
It is. Now, here's where it gets interesting. Activating the beta-1 receptor increases the heart muscle's contractility, so it makes it pump harder.
Okay, makes sense. But activating the EP4 receptor, even though it also increases CAMP levels, doesn't have the same effect.
Wait, what? But if they both increase CAM-P, shouldn't they both have the same effect on the heart muscle?
That's what you'd think, right? If CAMP acts globally throughout the cell.
So what's going on then? If it's not a global signal, then what is it?
This is where the concept of microdomains comes in. Think of it like a bustling city. Different districts specializing in certain activities.
Okay, so different neighborhoods within the cell.
Exactly. Microdomains, these are tiny localized regions where signaling molecules can be concentrated, creating these hot spots of activity.
So instead of CAMP being like a citywide announcement, it's more like targeted messages delivered to specific neighborhoods.
Precisely. And this could explain our heart muscle puzzle. Maybe beta-1 and EP4 receptors, even though they both increase CAMP, are doing so in different microdomains.
So different locations, different effects, makes sense. But how do these microdomains actually form? I mean, it's not like there are tiny walls inside the cell.
Yeah. Well, there aren't walls exactly, but there are these amazing proteins called AKCAPs, a kinase anchoring proteins.
AKA piece. Got it. Another acronym for the cellular vocabulary. And what do they do?
Think of them as the architects of his microdomains. They're scaffold proteins. They anchor another key player called protein kinase A, or pKa, to specific spots within the cell.
Okay, so they're like those velvet ropes that guide VIPs to their exclusive area in a club.
Exactly. And get this, different AKPs bind to different types of PKA. So you have this incredibly organized and intricate signaling network.
Wait, hold on. Different types of PKA too. This is getting complicated.
It is, but that's what makes the system so versatile. Over 50 types of AKPs, various forms of PKA. You have an incredible array of potential outcomes, all within a single cell.
Wow, so it's like a whole cellular dance floor with everyone in their designated spot.
That's a great way to put it.
But how do these AKPs actually bind to PKA? What's their secret handshake?
The key lies in this unique structural element called the amphipathic helix.
Amphipathic. That rings a bell, but I can't quite place it. Refresh my memory.
Imagine a magnet. One side attracts metal, the other repels it. An amphipathic helix is kind of similar. One side is hydrophobic, meaning it repels water, and the other side is hydrophilic, attracting water.
Right. I remember now.
Yeah.
But how does that help AKPs do their job?
Well, that hydrophobic side of the helix, it fits snugly into a specific binding site on PKA, like a key in a lock.
Yeah.
This makes sure PKA is anchored in the right place within that designated microdomain.
So it's all about precise molecular interactions.
You got it. And this idea, this principle of specific interactions, it's not just for AKPs and PKA. It's a fundamental theme in how cells talk to each other, you know?
Right. It's all about the right molecules fitting together in just the right way.
Exactly. And to see another example of this, let's shift gears a bit.
Okay, where are we headed?
Let's talk about thyroid hormone receptors.
Thyroid hormone. Okay. That's all about metabolism, right?
It plays a big role in a whole bunch of bodily functions: growth, development, energy expenditure.
So thyroid hormone receptors, those are the gatekeepers for those signals.
Exactly. And what's really cool about them is that they can act as both gene silencers and gene activators.
Hold on, they can do both.
Yep. It's like a molecular switch, flipping between those two roles depending on whether thyroid hormone is present or not.
So when thyroid hormone is around, they activate certain genes, and when it's not, they silence them.
Wow, that's pretty slick.
It is. But of course it gets even more complicated. It turns out TRs can also repress gene activity even when thyroid hormone is present, if
Yeah.
they bind to specific DNA sequences called NTREs.
NTREs. So those are kind of like throwing a wrench into the system.
You could say that. They add another layer of complexity.
Hmm.
The DNA sequence itself can actually influence how the TR behaves, even when the hormone is there.
Wow, so it's not just a simple on-off switch after all.
Nope. And there's more. TRs can also form dimers.
Dimers, like pairing up.
Exactly. They pair up with themselves or other nuclear receptors like RXR.
So they're like teaming up to get the job done. Right. And these pairings, they further diversify the effects of TRs. And then, to make things even more interesting, we have cofactors.
Cofactors. Okay, what are those?
These are proteins that interact with TRs. They can either enhance or repress their activity.
So it's like you got TRs, dimers, cofactors, and specific DNA sequences all working together to like fine-tune gene expression.
That's a great way to picture it. It's a complex system with a lot of moving parts.
A lot of moving parts, that's for sure. And I have a feeling there's even more to this story, right?
Oh, absolutely. But we'll have to save that for the next part of our deep dive.
Okay, I'm ready to keep going. My brain is definitely warmed up now.
Great. Well, let's go back to those cardiomyocytes. Remember those heart muscle cells we were talking about?
Yeah, with the beta-1 and EP4 receptors, both increasing KMP, but only one affecting the heart muscle's contractility.
Right. And we were just starting to get into microdomains and AKPs.
Yeah, those are fascinating. But I'm also curious about how biased agonism fits into all this.
Ah, good point. It could definitely be playing a role here, too. You see, different ligands, even if they both lead to KMP production, they might actually have different signaling biases when they activate those receptors.
Signaling biases, what do you mean?
Well, they might favor certain downstream pathways over others. You know,
Right.
it's not just about turning the receptor on, it's about how you turn it on.
Oh, I see. So even though both ligands activate the same receptor, they could end up having different effects because they're triggering different pathways.
Exactly. So, for example, you could have one ligand that activates the beta-1 receptor in a way that strongly favors the pathway that leads to increased contractility. But then another ligand, it might activate the same receptor, but it biases the signaling towards a different pathway, one that doesn't affect contractility at all.
So same receptor, different outcomes. That's incredible.
It really highlights how important it is to look beyond just whether a receptor is activated or not. We need to understand the nuances of how it's being activated and what specific pathways are involved.
So are there any real-world examples of how this biased agonism is being used?
Oh, definitely. It's showing a lot of promise in pain management, for example. You know how opioid drugs like morphine, they can be really effective painkillers.
Yeah, but they also come with a whole host of side effects.
Exactly. Respiratory depression, addiction, those are serious concerns.
So could biased agonism help us develop pain medications that are uh, safer?
That's the hope. Researchers are looking for opioid ligands that can activate the mu-opioid receptor, which is the one responsible for pain relief, but without activating those other pathways that lead to the nasty side effects.
So finding a bias key that unlocks pain relief without opening the door to all those other problems.
Precisely. It's a tough challenge, but there's been some promising early research.
Wow, that's really exciting. It seems like biased agonism could change the game for drug development in a lot of areas.
It definitely could. And allosteric modulation is another game changer. It's being used to develop some really interesting drugs, too.
Oh yeah, like what?
Well, there's some cool stuff happening in the treatment of anxiety and sleep disorders. Have you heard of GABA receptors?
Yeah, I think so. Aren't those found in the brain?
They are, and they play a crucial role in regulating anxiety and sleep. In fact, there are drugs called benzodiazepines that target GABA receptors, and they're commonly used to treat anxiety and insomnia.
Right, right. But I think those can also have some downsides, right?
Yep.
Side effects.
Yep. Drowsiness, dependence, those are definitely issues.
So allosteric modulation, could that help us develop drugs that target those GABA receptors more precisely?
Exactly. Instead of directly activating the GABA receptor like benzodiazepines do, allosteric modulators can fine-tune its sensitivity to GABA, which is the natural neurotransmitter.
So instead of just flooding the brain with calming signals, they're kind of adjusting the volume knob on those signals.
A perfect analogy, and this can lead to a more subtle and potentially safer effect. There are already some allosteric modulators of GABA receptors being used to treat insomnia.
Wow, so instead of just knocking someone out, these drugs can help them fall asleep without making them super groggy the next day.
Exactly. And the research is ongoing. They're looking at allosteric modulators for a whole range of receptors now, from the immune system to cardiovascular health.
It's amazing how far we've come. It's like we used to have these big blunt tools to try and fix things in the body, and now we're developing these really precise instruments.
It's a totally new way of thinking about drug development. And it's all thanks to a deeper understanding of these really intricate molecular mechanisms.
It's mind-blowing when you think about it. We've talked a lot about how biased agonism and allosteric modulation can help us create better drugs. But can you, I don't know, zoom out a bit for me? What does all this mean for medicine in the bigger picture?
That's a great question. It's not just about drugs. These discoveries are changing how we understand how cells work, how they communicate with each other.
It's like we're cracking the code of cellular language or something.
Exactly. We're learning how to send specific messages to cells and get specific responses. It's pretty powerful stuff.
You can see that. So what are some areas where this could really have a big impact?
Well, personalized medicine is a big one. Imagine a future where we can tailor treatments to an individual's unique genetic makeup, to their specific cellular responses.
Wow, that sounds like something out of science fiction. Is that really possible?
It's becoming more and more realistic as we learn more about these mechanisms. We're already seeing genetic testing that can identify people who are more likely to respond to certain drugs or to experience specific side effects.
So it's not just one-size-fits-all medicine anymore. It's becoming more personalized, more tailored to the individual.
Precisely, and this is just the beginning. As we delve deeper into biased agonism and allosteric modulation, we're going to find even more applications, I'm sure of it. Regenerative medicine, synthetic biology, the possibilities are really vast.
Wow, this is really incredible stuff.
Yeah.
But I have to admit, my brain is feeling a little bit full right now.
I understand. It's a lot to process. But I hope you're leaving with a sense of, I don't know, maybe a sense of wonder about how complex and elegant these cellular mechanisms are.
Definitely. It's amazing to think about all these tiny molecules doing these intricate dances inside ourselves, and that we're finally starting to understand their language.
And that understanding is going to lead to some really amazing breakthroughs in medicine, I think. More precise, more personalized, more effective treatments. It's an exciting time to be in this field.
It really makes you appreciate all the amazing things happening inside us all the time. You know, Things we don't even realize.
It is pretty incredible. It's like a whole world down there. All these signals, these feedback loops, these intricate
And
systems.
we're starting to learn how to actually control those systems,
Yeah.
to tweak things, to fine-tune the responses.
That's the power of this knowledge, right? We're not just observers anymore. We can actually intervene, maybe even fix things when they go wrong.
But that also brings up some big questions, doesn't it? I mean, if we're getting this good at manipulating things at the cellular level.
Oh, yeah, for sure. It's like they say, with great power comes great responsibility. We have to think about the ethics of all this.
Exactly. It's not just about the science anymore. It's about how we use that science, how we make sure it benefits humanity.
Right, we need to have those conversations,
Yeah,
about the risks, the benefits, and we need guidelines, regulations, making sure all these discoveries are used for good.
I couldn't agree more. It's a reminder that science isn't just about, pushing the boundaries of what we know. It's about using that knowledge wisely, ethically.
It's a group effort, really.
Yeah.
Scientists, ethicists, policymakers, everyone needs to be involved.
So as we keep learning more about cell signaling, about all this incredible potential of biased agonism and allosteric modulation,
We can't forget about the ethical side of things.
absolutely, it's a journey, a scientific journey, and an ethical one. And we have to be careful how we navigate it.
I think that's a great way to put it.
Well, this has certainly been an amazing deep dive. I'm leaving with, I don't know, a whole new appreciation for how intricate and elegant this whole system of cell signaling is.
And how much potential it holds, but also, yeah, the responsibility that comes with that.
Absolutely. It's mind-blowing to think that all this is happening inside us all the time. These tiny molecules, these molecular symphonies orchestrating life itself.
And we're just starting to understand the music.
It's incredible. Thanks for joining me on this journey. I can't wait to see what other secrets we uncover as we keep exploring the world of cellular communication.
It's been a pleasure. And remember, everyone, the learning never stops. Keep exploring. There's always more to discover.